For our final installment of this chapter, we're going to be looking at the final group of macromolecules, which are called nucleic acids. Now, you know nucleic acids, at least you hear about them all the time, particularly DNA and also RNA. So that's what they are. Nucleic acids are polymers of these monomers we're about to look at, and what they do is they hold information for everything that really makes us up. So this is the code in the DNA to all of the things which create us, all of our structures, etc. And that RNA is a part of the way that this code is interpreted. So here we go. We're going to find nucleic acids in a variety of places, but certainly in cells. We're going to look at the nucleus of eukaryotic cells, and then we're going to look at some other places where we can find nucleic acids. So DNA is the big one that people know mostly about, I think, and it is the coded instructions for all proteins. What we very often call the genes, our genes are components that we're going to find on the DNA molecule. So DNA has all the codes for everything. So within the 23 pairs of chromosomes that you inherited, one of those 23, a set of 23 was from your mother and one was from your father, we have the code for everything that makes you up. And so that code then needs to get translated into various proteins. And to do that, we need another molecule that is also a nucleic acid called RNA. So DNA is the code to all proteins. RNA translates that code into the actual string of amino acids, into that primary structure of a protein, and then the protein continues to fold and to what its actual shape would be. So let's take, so RNA, we said, is involved with taking the code from DNA and then ultimately making the protein. So there's three main types of RNA. There's messenger RNA, which is mRNA, which is something is in the nucleus of the cell. It has transcribed some of the RNA. In other words, it's made a copy of some of that RNA, DNA, and now it's taking it out of the nucleus in order that we can make or translate that into proteins. The area where that's translated into proteins are called ribosomes, and they're also called ribosomal RNA or rRNA. So we have mRNA messenger, tRNA transfer, and then the rRNA, which is the ribosomal RNA. When we look at them, both DNA and RNA are made up of monomers called nucleotides. And a nucleotide is made up basically of this. It's going to have a base which has nitrogen in it, so that's why it's called nitrogenous base. It's going to have a sugar component. In this case, it'll be a pentose. Remember, pent means 5, so it'll be a 5-carbon sugar. And then there'll be one or more of another functional group called phosphate groups. So we're going to look at DNA and RNA, but we're going to look at these bases, these different types of bases that are the monomers, and the different types of sugars that can also be a part of their structure. Now, in truth, we're going to kind of do this rather quickly because we'll look at it much more clearly when we go on to a future chapter. But we'll just start here with DNA. So when we look at DNA, DNA has this double helix structure, which looks like a spiral staircase. And on one side of you might think of it as the side of the exterior of this staircase, so not the rungs of the ladder that would be in between, but the outside of it, what we see are the sugars and the phosphates. So that's where we'd find them. The bases, the nitrogenous bases, are going to be in the inside. And there's going to be one on each of these strands, and then they're going to connect with hydrogen bonds. So there we go again with hydrogen bonding. Now these strands are called anti-parallel because they actually run in opposite directions. We'll take a quick look at that. So, whoops, that's right. So here are some of those bases. So here's thymine as one of the nitrogen bases. You can see nitrogen within it. Here's guanine as another one. Here's adenine, and here's cytosine. So one thing you might notice, just look at a glance, is that thymine is made up of this kind of a ring structure that is really a single ring, where guanine has a double ring. It's a fused ring. We have a 6 carbon, or 6, we'll just say, there's 2 different fused rings. And here's adenine, which also has 2 fused rings, and then cytosine, which is a single one. So we very often say A, T, C, and G. So those are the letter abbreviations for adenine, thymine, cytosine, and guanine. So you might also notice here's our hydrogen bonds, the dot, 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 and that we have thymine bonds with adenine. So A to T or T to A, these 2 bases or nitrogen bases are going to bond together. And then guanine and cytosine bond together. So G and C, or C and G, always bond together. And in truth, this part I'll have to explain a little bit more, but it says one strand runs 5' to 3', and the other 3' to 5'. So I'll explain this more in class. You can see it in the diagram. And so they're talking about this idea of being 5' here and 3' on this one. So in other words, if we're going 5 to 3, we're going this direction. And then we're going 5 to 3, but in the opposite direction on the one below it. So this is the anti-parallel part. Now, the prime number is really talking about an attachment point, and we'll see this in more detail coming up. So again, this is the breakdown of why DNA is so important. DNA has the code. That code, part of that, and you can see it here on this double helix molecule of DNA, which is what makes up our chromosomes and our nuclei of our cells. Some of this has information on it, so this could represent a gene that has to get copied by transcription into mRNA, and then mRNA will leave the nucleus, go out into the cell and the outer part of the nucleus, and help to translate that information into the chain of amino acids that we know is the primary structure of the protein. Well, then it will continue to fold and do secondary, which is the hydrogen bonds, and the tertiary, which is some of those R groups interacting, and then even quaternary in some cases, and forming the actual shape and structure of that protein. So DNA, RNA. Over here in this translation piece, we would also see where the ribosomes are doing this. So that would be the rRNA. So we're going to see that DNA has these genes, which is these sequences of information on them. RNA copies it, messenger RNA, and then it's going to bring it out into the cytoplasm to eventually get translated by the ribosomes. Now this part, I'm going to go into much more detail on this later on. So this is just a preview. So just understand that there are these 2 different types of nucleic acids, DNA and RNA, that are made up of these sugar phosphates and then then nitrogen bases, and that they form DNA. If we're looking at that double helix structure, that's what's in our makes up our chromosomes. And that RNA, there's three different, at least we'll talk only about three different types of RNA. And those eventually will make the proteins that we see. This gets into, again, that same, I've pretty much described this. What does RNA do? Okay, mRNA is a single-stranded copy of a gene. Ribosomes are called rRNA. And tRNA is what we're going to see when we get out into the nucleus, out of the nucleus. We're going to see what tRNA actually is. So just stay tuned on that. This is looking again, this is a preview really of what we'll get into more detail on. I'll talk about this in more detail in class, not just using these slides, but I think that's all we're going to really get into on these different nucleic acids. So this is kind of a nice chart though. It gives you an idea of if you were going to talk about DNA and its structure, and which would include this idea that it's a double helix. It has a sugar on it called deoxyribose sugar, and where it is in the nucleus and what it does carries genetic information, and that it has these nitrogen bases, cytosine, thymine, adenine, and guanine. RNA has also certain components that are similar, but is only single-stranded. In most cases, it's something that's made, some of it is made in the nucleus and sent out, and it has a little different sugar on it. Instead of deoxyribose, it has ribose sugar, and then it has these nitrogen bases. So you'll notice that that 3 out of 4 are exactly the same as the DNA, with one exception, and that is instead of thymine, we have a different nitrogen base called uracil. I'll talk about the purines and the pyrimidines. Don't worry about that right now. And then finally, this is a really important idea in what is very often called the central dogma of biology. And that's this idea that DNA carries information and that information gets transcribed, in other words, copied, and translated into something we know of as proteins. So that there are some changes that have been made in the central dogma, but one of the first people to articulate this and explain it was Francis Crick, and you may have heard of Watson and Crick, the discoverers of the DNA, the shape of the DNA molecule, and that they both won the Nobel Prize for that. So there you go, that's it, and we will get into more explanation in class. So stay tuned, that's it.